Hello, everyone. This is JJ with NCIMS, and I have Dr. Zava with us today from ZRT Laboratory, and he is one of our featured speakers coming up for the Grand Rounds event on March 12th. So thank you so much for taking a moment to be with us today, Dr. Zava.
Thanks for inviting me.
Very welcome. Dr. Zava is a biochemist and researcher who wanted to provide healthcare practitioners and patients with a deeper understanding of the role hormones play in wellness. So, Dr. Zava, what led you to get into this type of research specifically?
Well, it's kind of a long story, but it started with President Nixon saying that he was going to declare war on cancer back in the 70s. So that was a part of really kind of where I got started. I was in my graduate program at University of Tennessee, and we had to make a decision about what we were going to do in our research program. And so I decided that since Nixon had declared war on cancer, that there was probably going to be money there, and so I decided to do cancer research. I could have done brain research. I'm a scientist. I'm very curious about a lot of different things. And so got involved in that. So it was basically just general cancer research that I did. I was looking at a chemical called dimethylbenzanthracene, and it's a chemical in the environment that could, make people have cancer. But in particular, it was an interesting chemical, small structure that looked an awful lot like estradiol. And when you give it to rats, they get mammary cancers. They don't get any other kind of cancer, so they get cancers of the mammary gland. So it was a good model to look and follow. So from my graduate work, I then went to University of Texas in San Antonio and did a postdoc where we were digging in a little bit deeper, and they knew that I was interested in that particular chemical. They knew that and we used that chemical to induce cancers in rats, so we could study, because it's very similar to human cancer. So we were more interested in, okay, how does the estrogen stimulate the growth of the tumor? So that's really kind of where estrogen came in, and why is it the progesterone tends to be inhibitory? We were working with human tissues, but the experimental model was rats. And so we were extracting tumors to look at their levels of estrogen receptors. So that's kind of, we think of hormones on one side, and then the hormones are going into tissues and doing something. So estrogen was, we know was binding to a very specific estrogen receptor. It was triggering the receptor to go in the nucleus. We called it translocation, and it would turn on the gene. So it would turn on the gene for new estrogen receptor, turn on the gene for progesterone receptors, turn on the gene for growth, because that's what estrogen does. It's growth promoting. That's not necessarily a bad thing. It's a bad thing if it's a tumor. Good thing if it's the lining of the uterus that just shed, or the brain, or the replenishment of parts of the body. So, and we used a lot of cell cultures that had receptors in it. So I was sort of a receptorologist. I worked a lot with sort of what's in the black box, as opposed to, okay, here's the estrogen out in the bloodstream, and this is how much is there. I was looking at, okay, what happens when it binds to receptor and it goes in and starts to do things. So again, long story short, I then went to Switzerland and was involved in a large-scale international clinical trial in breast cancer. And we were starting to work with a drug called tamoxifen. Tamoxifen is a drug that it'll bind the receptor and it locks it up. It just paralyzes the receptor. So that's why it works. And so it slows the growth of the tumor. So what we learned is it's cytostatic, but it's not cytotoxic like a chemotherapy. So it'll stop the growth, but once you take it away, the growth can resume as long as some estrogen fits into that receptor. So my curiosity about estrogen was, well, gee whiz, what if you eat soy? What if you eat a phytoestrogen? Is that going to displace the tamoxifen? So I did a lot of research on that and published stuff and showed that, yeah, soy phytoestrogens and estrogens from the environment are capable of doing the same thing. They're capable of, binding that receptor. But that was just the model. It wasn't the whole human being. It didn't have anything to do with the immune system or the interplay of all the tissues with what's happening, at the tumor level. So I was involved in that clinical. I came back to the United States after about 5 years. So we're talking about 70s to 80s to 1970s, 1980s, and then was involved in several different groups and ended up in a place where I actually got a grant to look more at how some of these phytochemicals and other things might be displacing the tamoxifen from its binding site and reactivate tumor growth, perhaps, maybe, we didn't know. So that was what I got funded for. And part of that funding was linked to knowing how much estrogen was present. So had to develop an assay for estradiol. So there were other people doing that kind of thing, and so we're looking at estradiol levels. So I just developed. I said, okay, as part of the grant, develop a non-invasive way of monitoring estradiol levels in women. Saliva test. Okay, that's why I developed the saliva test, was specifically for that reason. So I developed that. Again, long story short, because I was running a breast cancer prognostic testing laboratory, working with pathologists, working with oncologists mostly, and then had this grant. And then everybody and their grandmother wanted to have their hormones tested. So I was good at doing that in a non-invasive way. So I could measure estradiol, I can measure progesterone. I had started that developing professional relationship with John Lee, who was starting to do, writing books about progesterone and his clinical experience, and also working with, the academic groups at the same time. So one thing led to another. I was working to develop more of the saliva testing, and so that part of what I was doing. So I was lab director both of the prognostic breast cancer testing lab and had written all the SOPs and standard operating procedures and everything to be able to do hormone testing, because we had to do this through the appropriate channels, CLIA, everything had to be done according to what they said you're supposed to do to commercialize the test. So we did that. And then there was less interest in the group I was working with for breast cancer, and they just wanted me to work on the saliva stuff. And I said, no, I'm not interested in that. So I moved. So I left there in 1997 in the fall. I just packed everything up, moved my family to Portland, Oregon.
Oh, wow.
And started a saliva testing lab. And my goal really was to continue and I have continued the research in breast cancer. So I've done a lot of work with people, written articles about, natural progesterone. That was the time when John Lee and I wrote the book on What Your Doctor May Not Tell You About Breast Cancer, because it was related to both estrogen and progesterone. So, progesterone's the great balancer. Estrogen, I've always said estrogen is the angel of life. It's very important for both men and women, but it could be the angel of death if you have too much of it, or it gets metabolized. It's like cholesterol. Too much of it, or if it oxidizes, then it's bad. Otherwise, it's present in every cell of our body. It's essential for health and life, just as estrogen is. So it's important that it's present at the right concentration. So that was really a lot of where I was coming from is that making sure that the estrogen, if women are having symptoms of estrogen deficiency or estrogen excess, that we had some method to be able to monitor it. And saliva is a very good way to do it. Blood spot is, urine, serum. We test all those types of body fluids. That's part of what we've commercialized. It's not like I'm telling you, oh, saliva is the only way to do it. That's not how I feel about it. It's saliva, like I said, saliva, blood spot, urine, dried urine, and serum. We do them all. They all work. Some work better than others for different reasons. So that was a part of what we developed with regard to estrogen. And then I was more interested in about 2006. I hired some people to start the work on looking at estrogen metabolites, because I was reviewing grants for the NIH on estrogen metabolism. So what we call catechol estrogens, and this is one of the subjects I know that the group is interested in me talking about. So we were buying mass specs and developing methodology. It took us probably 4 to 6 years. It's a lot of hard work, to develop the methodology to do it and get it right. And we were doing it in dried urine. So you could say dried urine. Some of the people that worked at ZRT added some other letters to it and called it something different. But it's basically the same thing, and it was developed at ZRT. So what we found, and we did at the same time, in about 2012, we were actively involved in a clinical research project with a Chinese doctor who was running this clinical study out of a group in China. So we looked at breast cancer patients. So we'd already developed the methodology to look at the catechol estrogens. So we, dried urine, we could look at the native hormones, estradiol, estrone, estriol, and we could look at the catechol estrogens. So basically, it was 2-hydroxy estradiol and estrone and 4-hydroxy estradiol and estrone. So the 4-hydroxys are the bad ones. They're the ones that are kind of like the oxidized lipids, because that's basically what they are. They're oxidized molecules, because all the hormones come from cholesterol. So it's sort of like the same thing. They get oxidized, and they become very reactive molecules, and they can damage DNA. And Ercole Cavalieri and Rogan were 2 of the people that did some of the really early work, and I was reviewing their grants. And so they got funded for that, and we became professional friends, colleagues, and so we were kind of talking about the same thing. So I wanted to develop those assays. So we did develop. I hired people, and it took us about 4 years to do it. And we did the clinical study, and the clinical study is published. So anybody wants to, they can look up my name and what I've published, and it's a study that was published in China where we looked at women who had breast lesions, and they did a biopsy of the tumor or the lesion. They didn't know what it was, and when it was cancer, then they had them collect a urine sample, and then they had a control group. So that was what the study was about. So we looked at the levels of hormones in those women, and what we found was that the women who had infiltrating ductal carcinomas had much higher levels of the catechol estrogens, and they went on after we did that and did a lot more work to show what it was, and pretty much what Cavalieri had said all along, and that is that the estrogen is being metabolized down a different pathway to make the 4-catechol estrogen. 4-catechol estradiol was absolutely the worst. It was the most toxic. So when they put them into tissue culture, they could see that, and they could see if they amplified the gene that would actually convert estradiol into 4-hydroxyestradiol. That was the event that was causing the problem. So we know that we've developed the assay for that. We look at, and I have to say that it holds up. Just because you make a bad, catechol estrogen does not necessarily mean you're going to get breast cancer. It just says, look, okay, it's like you've got oxidized cholesterol. You are putting yourself at risk of higher risk of developing, if it's oxidized cholesterol, cardiovascular disease. If it's 4-catechol estrogen, it's higher risk for breast cancer. And what I see, because I'm working with some people, physicians who've been doing this for a very long period of time. We've published. Rebecca Glazer, Dr. Glazer. She was a breast surgeon, and now is working with breast cancer patients. So we're looking at the levels of the catechol estrogens, and it's interesting because using aromatase inhibitors to lower the estrogen burden at the same time of using testosterone. When we see a tumor start to grow again, all the estrogens are low: estradiol, estrone, estriol, all the 2-hydroxys, because there's very little precursor estrogen. But what we see is the 4-hydroxy estrogen will be elevated. So there's no question in my mind, that is the one that we're going after. That's the one we want to see, because so that would be the bad news. The good news is there are lots of things you can do. You can number one, reduce the burden. It's like, how many bullets are coming out of a gun? I shouldn't say that, but it's like, okay, you reduce the number of potential hits that are occurring in the body. So just by reducing the estrogen, and that's diet. That's eating cruciferous vegetables. That's, taking certain things. Progesterone is an example, because it metabolizes the estrogen down beneficial pathways. So that's really the basis of what we're doing, and what I'm interested in continuing as long as I can last. You probably figure out my age. From the mid 70s, I was in graduate school. So I've been doing this stuff for a long time.
Long time. Well,
yeah.
I have a kind of an off the wall question for you. Is what did the 8 or 9-year-old David Zava think he was going to be doing when he grew up? And how did you get to become so motivated to go into this research?
I'm just interested in research. I honestly could have done anything. I was born a scientist.
So you were always a curious kid.
I'm not, a lot of people that do what I do, they do it for the money, they do it for the fame, they do it for, it's not really genuinely trying to help people as much as it is to help themselves. That's kind of disgusting to me. And I'm just a pure scientist. I'm very interested in science. I found, I was interested in estrogen because it's a hormone that, like I said, it's the angel of life, but it could turn on you if you're making too much of it, or you're converting too much. So I wanted, it's what keeps both males and females healthy. Testosterone more so for males, but definitely for females, it's kind of in a sense going back to what Robert Wilson said a long time ago when he wrote that book that was so controversial, that estrogen was beneficial. They were just trying to push, horse urine on women, and that didn't go over too well, because too many people that like horses, and they don't like the horse torture. So it's I've heard Alan Gaby say it's got the karmic fingerprint of horse torture. So And it really does. So people didn't like that. It doesn't mean it didn't work. It did work at the right concentration. And it, when they looked at the incidence of breast cancer in the Women's Health Initiative study, what they actually found was that women using a lower dose of Premarin actually had less risk for breast cancer. So it's not like it's causing breast cancer the way it's being used. I think they have to be. What I'm about now is how you keep the estrogen within a physiologic range, and what is the physiologic range for the body fluid that we're working with, and what how should it be balanced with progesterone? What's the best way to balance the estrogen? So it just people are healthier when they use. My mother used, she's passed now, but she used hormones her entire life until she died at 93. So it's they're good for you. People say, well, you should stop 5 years after menopause. Really? Serious? No. It's like, okay, you want to go through this degradation process, go ahead. I can't stop you, but it's not recommended. It's and some women can make enough on their own. That's not a lot of women, but guys they can't continue to make the healthy levels of testosterone. Women can't continue, and they won't make any progesterone. So if they use estrogen, they should be using progesterone along with it as a balance. And we know the benefits of progesterone. So and they, some who've had their ovaries removed probably should be thinking about a little bit of testosterone too. But it's not just those sex hormones that keep you healthy. It's thyroid, it's insulin, it's
Sure.
cortisol. All of those things are really important. So we measure those, and we can see, and we look at symptoms in people. We look at how much hormone they're taking. We look at how they deliver it. We have millions of data points now that are in our system, in our computer, and we look at that to see, okay, what's the how are things changing? What are the symptoms? And that's some of the stuff I'll talk about.
That's what I was just going to ask you. What can our folks look forward to on March 12th?
Yeah, I'll talk about, okay, what happens when you move past physiologic level, because quite frankly, when you get pregnant, the estrogen goes way past what physiologically. So you could say, well, that's physiologic too, but do you want to put yourself in an estrogen state for a long period of time? I would say most women probably would say no, but I love the way it made my skin beautiful and my hair nice. So because that's the pregnancy glow. What I'm talking. Heard of the pregnancy glow? That's estrogen. That's estrogen and progesterone and estriol. Estriol is actually higher than estradiol during that time, and that's protective. And I'm surprised that the powers that be want to ignore the benefits of estriol. In the breast cancer book I wrote with John Lee, I said that estriol plays an important role for lower reproductive tract function. So it's great for vaginal atrophy, vaginal dryness, because there are receptors there. See, I'm a receptor guy. There are receptors there in that tissue that respond equal to estradiol. So they have the same affinity. But when you get into the uterine lining or you get into the breast tissue, the estriol is only 1/10 as potent because they're different receptors. And the estriol doesn't work that well for brain function either. Estradiol plays a really important role in helping to regulate serotonin levels. And so, it inhibits monoamine oxidase, which is the enzyme that converts serotonin to its metabolite, inactive metabolite. So you can, by having estrogen, you're keeping the serotonin levels, a little bit higher. So that's the sort of, happy day, kumbaya, bluebirds on my shoulder, So that's
Okay, great. Well, we don't want you to tell all the secrets for the presentation that you're going to do on the 12th, but thank you.
That's the stuff I'm going to talk about. Yeah.
Thank you so much. And I just wanted to also mention to folks that are watching, if they happen to have missed the registration period that's free for members, or if they're a non-member of NCIMS, they can reach out to Greg at ZRT Lab for a special promotional code to give them a discount off of their registration. And Greg will have that ready for them if they want to join us on March 12th.
Right.
Okay, so folks can go to ncims.com to register, and if they have any questions, reach out through the website. So thank you, Dr. Zava, again for your time.
All right, thanks, JJ.
Okay.
Appreciate the time. Take
You
care.
as well. Okay,
Bye-bye.
bye.